Hello and welcome to Biology 105, an introduction to microbiology. We're going to be looking at lots of little critters, and we're going to be focusing on the ones that might cause you some harm. There's a lot to cover here, so let's get started. I'll give you a quick overview of what we're going to look at in today's topic. I'm going to start with an introduction to microbiology. So what is microbiology? What are we going to look at in this course? Next, I'll talk about why microbes are important to us. We're going to focus on pathogenic microbes, ones that can cause us harm. And of course, those are important things to study. But I also want to point out that the majority of microbes are not harmful. I would feel rather bad if you went away from this course thinking that all these little invisible things around you are dead set on killing you. That's not the case. There are many microbes that are essential to life. We wouldn't be here without microbes. Next, I'll talk about the history of microbiology. I'll talk a little bit about some of the people that came up with important discoveries and important techniques that affected how we study microbes. Also, I'll talk about the impacts that pathogens have had on human history. We'll talk about the plague, for instance. And of course, we'll talk about what's going on right now with COVID-19. Finally, I want to introduce you to the scientific method. And the reason for that is we're going to talk about some rather important experiments throughout the course of the course. And we're also going to do some experiments and do some hands-on testing in the lab. So it's important that you understand how the scientific method works. Let's start with the fundamentals. What is microbiology? Well, micro means small. Biology means the study of life. So you might think that microbiology is the study of tiny little living things, and you'd be mostly correct. So bacteria are microscopic. Microscopic means they're too small to see with the naked eye. Definitely, those are microbes. But viruses are also considered to be microbes, but they're not alive. Viruses are capable of reproduction, but they can't do it on their own. They have to trick a cell into making copies of their genetic material and producing their proteins and other components that make up a virus. We'll look at smaller things as well. We're going to look at proteins and the amino acids that make them up. We'll look at DNA. We'll look at organelles within the cells, and we'll see how pathogens manipulate that material. On the larger end of the scale, we're going to look at some eukaryotic, single-celled pathogens that are just barely visible. We're also going to look at ticks and mosquitoes and other insects and arthropods that can carry pathogens from one organism to the other. They serve as what's called a biological vector. In addition, we're going to talk about some multicellular parasites that you can definitely see with your unaided eye. Things like tapeworms and Ascaris, which is an intestinal roundworm, and pinworms and hookworms and all sorts of worms, and a few other things. Why are we including that? Those are not microbes. Well, they are infectious. They do cause infections. They're fascinating, at least I think they're really fascinating. And also, you're not going to be exposed to those large parasites, academically speaking, in your other courses. So it makes sense to kind of fit that in here. Okay, so the course covers a broad scale of organisms and almost organisms. That's what you're seeing here. We're covering a lot of stuff. Now pay attention to the ruler there. You'll see that each notch on the ruler, the size of that measurement increases by a factor of 10. This is a logarithmic scale. So we have 1 millimeter, we go up to 10 millimeters, we go up to 100 millimeters. So again, each division on this ruler is a factor of 10 increase. Now you're probably familiar with millimeters, and I hope you're familiar with meters, but you might not be familiar with micrometers. Now we're going in the other direction, we're getting smaller. 1 micrometer is 1/1000th of a millimeter. If we want to go even smaller, we have nanometers. 1 nanometer is 1/1000th of a micrometer. And we could get smaller. We could go to picometers, PM, not shown here, but 1 picometer is 1/1000th of a nanometer. When I'm talking about microbes in this course, I'll quite often be using the unit micrometer. Micrometer consists of the Greek letter mu, which is a U with a little tail on it, followed by an M. So make sure you understand what that unit means. Again, it's 1/1000th of a millimeter. If we're looking at molecules, if we're looking at viruses, I might use the unit nanometer, NM. Again, 1 nanometer is 1/1000th of a micrometer. So be able to convert between those different units. When we're looking at microbes, we can use a compound light microscope, and that's what we'll use in the lab. Our microscopes will magnify up to 1,000 times, and that's enough to look at bacteria. However, if we want to see more details, we need to use an electron microscope. We have scanning electron microscopes, which allow us to see a 3-dimensional image of the surface of whatever it is we're looking at, and also transmission electron microscopes that allow us to see even higher magnification and see internal details. We'll come back to these different tools when we talk about the tools we use to study microbes. As many of you probably know, my background is not in medical science. I have a more academic biology background. My focus has always been comparative biology and evolutionary biology. And if I was teaching this course to a group of biology students, not individuals that are going on into the health sciences and medical sciences, I would be telling you how wonderful all these organisms are. We'd go through them one by one, and I'd point out all the things that make them really cool and make them different from other organisms. You guys, of course, are going into the health field. So your main interest is, how do we kill these things, or how do we limit their growth? Well, I'm going to take a similar approach. I'm going to tell you what makes all these creatures incredible, what makes them special, and then we'll figure out how we can use those special characteristics to kill them. So in this course, we are going to be looking at pathogens mainly, but again, remember that the vast majority of microbes are not pathogenic. We're going to look at different critters, and I'm going to tell you how we can limit their growth or get rid of them, and how we can diagnose the problems that they might cause within your body. With an understanding of the biology of pathogens, we'll be able to look at hard-pressing questions, like, what's the harm in double dipping? Now, you may or may not be a fan of Seinfeld, but if you are, you might remember the episode where George Costanza took a chip, dipped it in some dip, took a bite, and then dipped it again. And the Seinfeld show coined this term that we all use now, double dipping. There have been a number of studies on double dipping, and they've shown that on average, if you do this, you transfer about 2,000 bacteria from your mouth to the dip. And then, of course, someone else can pick up those bacteria. But is this a problem? 2,000, that's a big number. However, there are over 5 billion bacteria in your mouth. And there's over 700 different species of bacteria in your mouth. So that 2,000 that you pick up is a tiny fraction of what's already present in your mouth. Bacteria compete with each other. Bacteria have a hard time becoming established on a surface if there's already bacteria there doing their thing. So for bacteria, unless it's something very unusual, something pathogenic that you don't already have in your mouth, and it's something that's very virulent, it's able to spread and reproduce very quickly, bacteria probably aren't going to present a big problem. But what about viruses? Well, viruses are quite different. Viruses are not going to be competing with the bacteria that are in the mouth. Instead, the virus will penetrate the cells lining the mouth, so the cells that make up the epithelium, reproduce within the cells and cause damage. If the viruses are able to sneak past your immune responses, then they may become established fairly quickly and cause issues. But there's still other things we have to worry about. What about the dip itself? Viruses create big problems inside your body, but they're actually very, very fragile outside of your body. And the pH and chemistry of the dip might be enough to destroy that virus. So you can see that questions like this are difficult to answer. And we'll see that quite a lot in this course. So as we've discussed, microbiology is the study of tiny living things. We're also looking at some tiny non-living things, and to round out the course, we're going to throw in some macroscopic parasites. In no particular order, let's take a look at the diversity of creatures and non-living infectious agents that we're going to be looking at. We have bacteria, and the example here is Escherichia coli, E. coli. This is a bacterium that's found in your intestine, and it's generally quite helpful. It helps you break down your food a little bit more, and it releases vitamins like vitamin K. However, there are some strains that can be pathogenic, and of course there are numerous bacteria species that can be pathogenic. We'll look at fungi. Fungi can grow in one of two forms. Sometimes it consists of a chain of cells, and sometimes it consists of isolated free-living cells. If it consists of isolated cells, we refer to that as a yeast. And yeast, of course, can colonize parts of the body. It's not uncommon to find it in the mouth and other areas. Sometimes it causes problems, sometimes it doesn't, as we'll see. We have chains of cells that can live on the skin and cause conditions like athlete's foot. Then we'll look at protists, and protists are single-celled eukaryotic organisms. So a eukaryotic cell has a nucleus and it has organelles. What you're seeing here is Giardia, which is a little free-swimming critter that you can obtain from drinking water. You obtain it as a very resistant cyst, as we'll see, but it will swim around in your intestine, latch on to the inside of your intestine, and cause explosive diarrhea, something known as beaver fever. We'll talk about animal parasites. An animal is a multicellular organism where food is acquired through ingestion. So animals are multicellular, they don't produce their own food through photosynthesis, and they don't simply absorb small nutrient molecules the way that a fungus does. So we are animals. A tapeworm is a good example of an animal parasite, and we'll talk about a number of different worms that can cause you issues. We'll talk about viruses. What you're seeing here is the influenza virus. Again, these are tiny, tiny little structures. They're not technically alive, although depending on who you talk to, you might get different answers to that question, but they're not technically considered to be alive by most biologists. They can only be visualized with an electron microscope. We can't see them with a compound light microscope. We'll look at prions, which are even smaller, and they are simply proteins. They're proteins that can grab onto proteins on your neurons and change their shape. They actually manipulate the protein so that it looks like them. It's a pretty odd way of reproducing. But of course, when it does that, it damages the function of the neuron. Microbiology is a huge field with lots of different subdisciplines. So if we wanted to, we could divide it up further. You could take entire courses on each of these subdisciplines. Bacteriology is the study of bacteria. Virology is the study of viruses. Mycology is the study of fungi. Phycology is the study of algae. Now, algae are photosynthetic. They can make their own food, so they are not parasitic, so we're not going to be talking too much about them. We have parasitology, which generally refers to larger parasites, animal parasites, immunology, genetics, etc., etc., etc. And we're going to touch on all of these different subdisciplines. Just to further impress upon you the significance of microbes, I'm going to throw some really big numbers at you. There are more microbes in one spoonful of soil than the number of people that have ever lived. That's a pretty impressive number. There are at least 9,000 species of microbes that we have characterized, identified, and named. However, there's almost definitely many, many more, probably several million. When we're identifying microbes, like bacteria, things are rather difficult because the microbes look very similar. We can have two bacteria that look identical under the microscope. And if we want to figure out whether or not they're different species, we have to look at things like DNA sequences. And that's work that still has a long way to go. The other thing that's interesting about microbes like bacteria is that different species will quite often exchange bits of information. They'll exchange bits of DNA, which is not something that usually happens between species. So it becomes an interesting philosophical question as to what a species is. An interesting topic that we will come back to. There are probably upwards of 1,000 species of bacteria living in your intestines. In fact, you have more bacterial cells in your body than you have human cells. You have about 100 trillion human cells that make up you, but you have 10 times that number of bacteria. Think about that. Are you a human or are you a giant walking incubator, a giant petri dish for the bacteria that live on and inside you? There are an estimated 5 times 10 to the 30 prokaryotes on Earth. A prokaryote is an organism that doesn't have a nuclei or organelles. So bacteria, of course, and another group known as archaea that we'll touch on briefly. In fact, if you were to take all the life on the planet, trees and cheetahs and coyotes and insects, etc., giraffes, I don't know, whatever your favorite critters are. If you were to take all the life on the planet, gather it up and weigh it, you would find that the vast majority of that living goo that forms a layer over our planet is made up of prokaryotic cells. That number, 5 times 10 to the 30, is a big number. If you're studying microbes, that would make you a microbiologist. What does a microbiologist do? Well, this is a huge field. It covers a lot of different things. So of course, we have some microbiologists that are interested in pathogens. They're identifying pathogens, perhaps from samples of hospital patients, or they might be coming up with treatments and preventions and developing vaccines, something we'll talk about later in the course. We have a number of microbiologists that are food scientists. When food spoils or goes bad, what's happening is bacteria are breaking down the food and turning it into waste products that smell bad and taste bad. So a number of microbiologists are involved in the development of preservation techniques and additives that can be added to the food to slow the growth of bacteria. Fermentation is a process that can be performed by a number of different microbes. The best known example of this would be what yeast do. So yeast will convert sugar into ethanol. And that's where the alcohol comes from that's found in wine and beer and champagne, etc., and vodka and other hard spirits. Microbiologists might be using bacteria and other microbes to manipulate DNA from non-microbial sources. So for instance, in my research, I was isolating DNA sequences from lizards and reptiles and all sorts of different things. I was isolating these genes, putting them into bacteria and tricking the bacteria into making copies of that DNA so I'd have more of it to study. Also, I was tricking the bacteria into decoding that information and turning it into a protein so I could study the proteins as well. There are a number of research labs that are looking at all sorts of different areas of biology that have bacteria cultures that they use to do this. They use them to copy and express genes. But of course, there are some microbiologists that are interested in the genetics of the microbes themselves, and they're studying the cell biology of the microbes. There are a lot of people that study the ecology of microbes because microbes play a huge role in ecosystems. Other people study the evolution of microbes, and we're going to talk about how some of the different groups of microbes are related to each other. Geobiology is an interesting field. This is a fusion of geology and biology. Microbes play a huge role in recycling elements within the ecosystem. So recycling iron and sulfur and nitrogen, etc. They can also play a role in breaking down rocks. So they play a role in the recycling of the material that makes up the earth. A number of microbiologists work in the agriculture field. So let's say you've got a large farm, you've got a lot of cows, they're producing a lot of manure, a lot of waste. You don't want that waste washing into streams and rivers. If it does, what will happen is it provides food for bacteria and algae. They explode in numbers and end up extracting all the oxygen from the water and killing any fish that might be there. Instead, you can use cultures of bacteria to break down all that nutrient-rich material before it gets off the farm. There are also a number of microbiologists that are interested in pathogens that affect livestock and that affect crops as well. A lot of the pharmaceuticals and drugs that we use are produced by microbes. For instance, insulin. You can imagine that if you're diabetic and you need insulin, you're probably not going to get it directly from another human. We can't really go out and collect insulin from humans. At least we can't collect enough to use on people that require it because they're diabetics. In the past, what people did is they extracted insulin from horses and used that on humans, and it worked, but it's not the easiest thing to do, and also it's horse insulin, it's not human insulin. It doesn't work quite as well in a human as it could. So instead, what people have done, and this was first done decades ago, is they've taken the gene for human insulin from human cells, inserted it into bacteria, tricked the bacteria into making copies of it, and also using that information to make human insulin protein that can then be purified and injected into diabetic patients. Bacteria and other microbes can also be used to produce biofuels. So I mentioned that yeast can produce ethanol. We can use that as a fuel. There's bacteria that can produce isopropanol, butanol, acetone, a whole bunch of different compounds that can serve as important fuels or can serve important functions in industry. We can use bacteria in bioremediation. Let's say we have an oil spill. There are bacteria that will help break down that oil. There are bacteria that will help clean up sewage as well. And there are bacteria that can break down rock and release valuable metals. This is a rather new field known as microbial mining. But microbes do more than just supply nerds with jobs. They are essential to life on the planet. So let's start with those ones, those really important microbes. And hopefully you'll come to understand that, microbes are pretty darned important. A number of microbes are photosynthetic. That means that they can take energy from sunlight and use that energy to make their own food. What they're doing is they're taking carbon from carbon dioxide that's dissolved in the water around them. They're taking carbon, ripping it off of the carbon dioxide, sticking it together to make organic molecules. Also, there are a number of microbes that can fix nitrogen. What that means is they take nitrogen out of their surroundings and convert that nitrogen into ammonia and other compounds that are readily available to other organisms. What you're seeing here is cyanobacteria. You're seeing chains of individual cells. The larger cells are responsible for fixing nitrogen. We find these cyanobacteria in ponds and lakes and rivers and in the ocean, and they've produced most of the oxygen that we breathe. What's really quite fascinating is these bacteria popped up over 3 billion years ago, And they eventually gave rise to the chloroplasts that we find in plants and algae. So cyanobacteria make multicellular animal life, us for instance, possible by producing oxygen. Microbes also form the basis of just about every food chain. So we have here diatoms. These are single-celled organisms that are really quite beautiful when you look at them under the microscope. They're covered by a skeleton that's made of silica, glass basically. So inside each one of those shapes, we have one cell, and then there's two halves or valves made of glass that surround and protect that cell. If you have a piece of chalk, and I'm talking about the chalk that you would use to write on the blackboard, and it has to be kind of old school chalk. It's actually made up of diatoms. As you drag it across the chalkboard, what's happening is it's breaking down, it's releasing these tiny little microscopic particles, and each one of those particles is one of these little glass-covered microbes. And if we didn't have bacteria, we would be up to our necks in dead birds and dead giraffes and dead cheetahs, etc. Because one of the most important roles that microbes play in the environment is they act as decomposers. They take those dead animals and they break them down into smaller bits, into molecules that can be recycled and reused. They play a big role in nutrient cycling, especially nitrogen, carbon, and sulfur. And we'll talk about the nitrogen cycle in a bit more detail in just a second. We also enslave and harness microbes to do a lot of dirty work for us. We can use them to make dairy products, to make alcohol, to make other chemical compounds. We can use them for treating sewage. We can use them for bioremediation. And that's what you're seeing in the top picture here. So we've got an oil spill, and onto that oil spill, these guys are spraying a solution that contains bacteria that can break down that oil. We can use bacteria to clone genes, and what I mean by that is just to make copies of genes, and they can be genes from anything. We can use bacteria to make antibiotics and pharmaceuticals. There's a number of microbial eukaryotes that are used for that as well, yeast most notably. And we can use microbes for industrial processes. The nitrogen cycle provides an excellent example of the importance of microbes in the environment. There is nitrogen all around us. The air that we breathe is 78% nitrogen, but the nitrogen in the atmosphere is held together as diatomic molecules by triple bonds. We have two nitrogen atoms stuck together very, very strongly in a bond that we can't break. We need nitrogen to make amino acids that we're going to use to make proteins and to make nucleic acids. We need the nitrogen to make the nitrogenous bases of those nucleic acids. We cannot get the nitrogen from the air because, again, we can't break those triple bonds. So instead, we have to get it from our food. There are nitrogen-fixing bacteria within the soil that can take nitrogen out of the air. They can break those triple bonds and take that nitrogen and build ammonia out of it. Ammonia can then be used by plants. Some of these nitrogen-fixing bacteria also live within little nodules in the roots of legumes, things like soybeans. So we have nitrogen from the air being incorporated into ammonia, and then there are other bacteria within the soil that convert that ammonia into nitrites and nitrates. Nitrates and nitrites are even more readily absorbed by plants. In almost all locations on the Earth's surface, the key thing that limits plant growth is the availability of nitrogen. In almost all ecosystems, plants never grow as big as they could because they never have as much nitrogen as they want. And if you fertilize your lawn, what you're doing is you're adding nitrates and nitrites, and then of course you get a much lusher lawn. Also within the soil, we have bacteria known as denitrifying bacteria. They do the opposite of what nitrogen-fixing bacteria do. When you fix an element, you take it out of the air typically, and you use it to build bigger organic molecules. Denitrifying bacteria take nitrates, break them down, release the nitrogen into the air where it becomes a diatomic molecule again and becomes inaccessible. So let's say you're a farmer and you're growing some crops. Well, every year when you extract your crop, you're going to remove nitrogen. And that was nitrogen that came from the air originally, but was incorporated into the soil before the plants took it up. You might every so often throw in a crop of soybean because that will help the incorporation of nitrogen into the soil. The other thing you're probably going to do is you're going to till the soil. You're going to turn it over with a plow. That gets rid of weeds, but also what it does is it exposes denitrifying bacteria to oxygen. And denitrifying bacteria are poisoned by oxygen. They are anaerobic bacteria, something we'll talk about later. And that's going to decrease the loss of nitrogen from the soil. Now I should mention that there's lots of other bacteria and also fungi that are involved in this process as well that are going to break down those plants and return that material, return that nitrogenous material back into the soil where it can be broken down into ammonia again. We can also manipulate microbes. We can use microbial cultures, for instance, to produce some rather tasty food items. With cheese, what you do is you take milk and you add bacteria to that, specifically lactic acid bacteria. These are bacteria that generate lactic acid as a byproduct. That acid causes the milk to curdle and become lumpy, and then you squeeze out the excess liquid and you have cheese. We could also add some fungus to the cheese. If you add penicillium, you get blue cheese, which is kind of an acquired taste. It's something I love, but I suspect a lot of you don't share that love with me. It's quite fascinating when you think about it. We talked about this in anatomy and physiology, but milk is actually modified sweat. You're taking that modified sweat, you're adding in bacteria to make it go bad, and then you're maybe adding some fungus to make it more flavorful. Cheese and also yogurt are the product of bacteria that acidify milk and cause it to curdle, cause it to become more thick or viscous. Bread is also generated by a microbe. It's generated by yeast. Remember that yeast is a single-celled fungus. There are several different species of yeast. What the yeast do is they take sugar and other nutrients and they break that down and release carbon dioxide as a byproduct. The carbon dioxide is going to cause the bread to rise, so we have bubbles of this carbon dioxide that get trapped in the dough. Of course, another byproduct of fermentation in yeast is ethanol. So ethanol is found in wine and found in distilled spirits like vodka, of course, and also in beer. Beer, champagne, sparkling wines also contain quite a lot of carbon dioxide, and that's what produces the bubbles. Now, if you're wondering, the reason you don't get drunk off of bread is because baking that in the oven evaporates all of that ethanol. Hopefully, I've given you an appreciation of how important microbes are. Now, as a biologist, I tend not to use terms like good and bad. They're kind of subjective. I don't think there are good microbes and bad microbes, so to speak. They're all just trying to get by, just like we are. But of course, there are some microbes that cause us harm, and there are some microbes that benefit us immensely in many cases. What we're going to do now is we're going to look at relationships between microbes and between microbes and other organisms like ourselves. Symbiosis is a term that refers to close relationships between organisms. It means living together. It's a biological relationship. And we have three broad types of symbiosis that we're going to look at first. We can have two organisms that have a relationship where they both benefit. That's a case known as mutualism. And I really like the little fish down here, you can see we've got two happy fish. They're both benefiting. They're both very happy to be in this mutually beneficial relationship. Then we can have a relationship where one organism benefits and the other is unaffected. It doesn't benefit, but it's also not harmed, and that's known as commensalism. Next, we have parasitism, and this is where one organism is very clearly harmed. The other organism benefits. Let's take a look at some examples, microbial and non-microbial. So first of all, an example of mutualism. What you're seeing here are clownfish. Clownfish are marine or ocean fish that live very, very close to sea anemones. Now, sea anemones kind of look like plants, but actually they're animals. This is a colony of lots and lots of little tiny animals. They're actually related to jellyfish. And these little animals produce these stinging barbs. If something comes too close to them, they will stick out these little harpoons, these microscopic harpoons that will sting that organism. Clownfish are immune to this. They have a thick layer of mucus that helps protect them, but they also do something for the sea anemone. They wander around and they pick off little crustaceans and other things that might feed on the sea anemone and parasitize the sea anemone. So the clownfish gets protection. It's got a nice, safe place to retreat to, and the sea anemone has its parasites and other organisms that might harm it picked away. On and within your body, you have this natural collection of microbes that make up their own little ecosystem. We refer to these microbes as quote-unquote good microbes because they're not harming you, and in fact they're helping you. Having bacteria present on and in your body makes it far more difficult for competing bacteria that might be harmful to gain a foothold and get established. We often refer to these bacteria as microbial flora, and I should mention that that's kind of a misnomer. Flora means plant. These are not plants, but this term goes back hundreds of years to a time when we didn't understand what bacteria were. Some of these normal microbial flora produce vitamins that are quite important to your overall health. So it is important to have an established community of microbes on and in your body, and you can supplement these microbes by taking probiotics. An example would be bacterial cultures that are contained within yogurt. Yogurt contains Lactobacillus acidophilus, which is a bacterium that produces lactic acid. That's rather important in some parts of the body because that lactic acid will create a very low pH that inhibits the growth of yeast and other fungi that might cause you problems. E. coli, as we've discussed before, live within your intestine and they produce folic acid and vitamin K, and they also help break down your food a little bit further. The last example we looked at was a case of mutualism because both of the organisms in the relationship benefited. So remember, we had bacteria living on or inside a human, and those bacteria were given a warm, safe, moist place to live that also had a lot of food available. The human was receiving some protection from those bacteria. Now we're going to look at commensalism. This is where one organism benefits. The other organism doesn't benefit, but it's also not harmed. So in this example here, we have this rather interesting fish known as a remora that attaches itself to sharks and also to sea turtles. This fish has a suction cup on the top of its head, and it clamps onto the surface of a shark with that suction cup. It's a rather lazy fish. It just follows the shark around. It gets a free ride, but the shark, of course, will eventually catch something to eat. Sharks are rather messy eaters. They'll clamp onto something, they'll shake it back and forth, and in the process, a lot of little scraps of food will be thrown free. The remoras pop off the shark, they eat those little scraps, and then they hop back on the shark, awaiting their next free meal. So the shark is not really harmed by these remoras. unless it has a ton of them, which might create some drag, but really it's not a big hardship for the shark. But the remoras, of course, are very obviously benefiting. The example of commensalism that you're seeing here might gross you out a bit. In fact, you might wish I'd never told you about this one. What you're seeing are eyelash mites. So the photograph in the bottom right shows an entire mite sitting on top of the skin. Notice that it's got 4 pairs of legs. It's got 8 legs, which makes it an arachnid. It's related to other mites and also more distantly related to spiders. We can see in that photograph that we have 2 eyes, tiny little eyes. I'll highlight them here at the front. We've got this very long abdomen. And what they'll do is they'll burrow into a hair follicle until only that abdomen sticks out. That's what you're seeing in the photograph on the left. So we have this large hair shaft. That's the shaft of the eyelash. And then surrounding that, you can see the butts or abdomens of these mites sticking out above the surface. Their heads are burrowed into the oil and dead skin at the base of that pit. So you have sebaceous glands at the base of the hair shaft that are constantly producing this oil that coats the hair shaft, and in this case, feeds those mites. So those mites get a free place to live, and they get a free source of food. In most people, there's no harm done. A few people will suffer from allergic reactions and might get a rash, but that's fairly rare. And about 50% of people have these eyelash mites. You have mites on other areas of your body, and in fact, you have 2 distinct species of mites that live just on your face. The photograph that you're seeing in the upper right is the face of one of these cute little creatures. So again, the mite is definitely benefiting, but you're not, and until just a minute ago, you might not have even known that these things were on you. Next, let's take a look at parasitism. This is where one organism benefits and one is harmed. And this relationship is going to form the basis of much of this course. We're going to be talking about pathogens, and of course a pathogen benefits from a relationship it has with a host, but it harms the host. A lot of ecologists would consider parasitism a special case of predation. Predation is where one organism consumes another. We could be talking about carnivores, one animal eating another animal, or we could be talking about herbivory, where an animal eats a plant. That's also a case of predation. The difference is that in parasitism, the animal is attached to another creature, and it typically is slowly withdrawing resources from that creature. There are a number of very cool and rather gross examples that we could go into, and I promise you, we will get to some very gross stuff. But here's just a few that I picked out. In the upper left, you can see that we have an ant. And this ant has these very odd looking structures. They look like antennae, but they're not. This is an ant that has been parasitized by a fungus. So the fungus is going to be breathed in as spores by the ant. The fungus will germinate within the ant and develop and spread to the nervous system and change the behavior of the ant. The ant will climb up. It will be attracted towards sunlight, and it will climb to the top of a branch, to the top of a blade of grass, whatever, and it clamps its jaws down. And then what happens next is this fungus will rapidly divide and produce these structures that will give rise to new fungal spores. So we have this stalk that's coming out of the head of the ant, and at the end we have these structures called sporangia that will release fungal spores into the wind. That was the whole point of getting the ant up high, so that this zombifying fungus can spread to other ants. The other photograph at the top there on the right shows a fish that has an isopod, a type of crustacean, living within its mouth. This crustacean latches onto the inside of the mouth of the fish and devours the tongue. It removes the tongue of the fish and replaces the tongue, which is really kind of creepy. It connects to the blood supply of the fish and it sucks blood out of the fish for the rest of its life. Down the bottom, we have a roundworm that's living within a grasshopper. And I'll show you some videos of these later. They can be kind of disturbing. Believe it or not, that worm came out of that grasshopper. These are worms that will live within the grasshopper. They'll develop very slowly. They will very carefully consume different parts of the grasshopper that won't kill the grasshopper. And then when the grasshopper is attracted to water later in life, and this is something that's directed by the parasite, this worm will pop out, end up in the water, and it actually breeds within the water with like-minded worms. Let's take a look at some human examples. In the upper left, we have a tapeworm. So tapeworms are highly simplified animals. They lack a digestive system, they lack a nervous system, they don't have specialized sense organs like eyes, they don't need any of that stuff. They're living in the perpetual darkness of your intestine, and they're living in pre-digested food. You've done the work for them already. All they have to do is soak that stuff in through their skin. This particular tapeworm, you can see, is shedding segments, and those segments contain millions of eggs. So those eggs are going to be passed in your feces, and they might be picked up by another host. On the upper right, we have a louse. Lice are specialized insects that live on the outside of your body. They're ectoparasites, as we'll talk about, and they draw blood. We have 3 distinct species of lice that live on us. Down the bottom left, we have Ascaris, which is a type of roundworm that inhabits the small intestine. About 1/5 of the human population has these worms. So about 1.5 billion people contain these parasites. In the bottom right, we have Giardia. Again, this is a single-celled protist, a eukaryotic cell. It kind of looks like it's smiling at you defiantly. There's 2 nuclei in these guys, and then there's a slanted little vacuole underneath that. But this is a parasite, even though it's microscopic. It's attached to the inside of your intestine and it's drawing resources from you. Now again, I think I mentioned this before, but I'll mention it again. A parasite, technically speaking, is anything that causes harm to a host. So the host is not benefiting, the parasite is. Quite often in biology and medical science, though, when we're talking about parasites, we quite often are referring to multicellular parasites. Parasites have evolved some very elaborate reproductive strategies. There's 2 broad categories here when we're talking about parasites. Some parasites will stay put for a long period of time. Some will reproduce very quickly and then move on. So for many parasites, the chances of them finding a suitable host and getting established is very, very slim. And they will stay with the host in some cases for their entire lifetime. So with the worms that I talked about, if they're lucky enough to find their way into your intestines, they're going to stay there. And you might have a pet tapeworm for decades. And the tapeworms will release lots and lots of eggs. The eggs are released in the feces, and perhaps just a few of those eggs might find their way to another host. These types of parasites tend to cause minimal problems. If you have one tapeworm, you might not even notice. If you have a bunch, of course, you will. Other parasites reproduce very, very quickly and may cause a lot of damage and then move on to another host. And this is something that we're more likely to see in microscopic parasites, in microbes. I apologize for the really kind of lame comic here, but if you can't read it, we have a parasite coming in for a job interview because, of course, that's what they do. And this parasite is asking, what kind of openings do you have? Do you get it? There's one more situation we should address. What if both organisms are harmed? Well, this is competition. So we have two organisms that are competing for resources. They might be competing for space, nutrients, oxygen. If we're talking about trees in a forest, for instance, they might be competing for light. Is this a case of symbiosis? Well, again, it depends who you talk to. Is this a relationship or is this an anti-relationship? Well, either way, this is important. We talked about this already, but bacteria in your body that aren't causing you harm actually do help you. They help you because they compete with other bacteria that might cause you harm. There's this ecosystem in your body, and all of those organisms are fighting for real estate. Here's an obvious example of competition. We have two large cats that are competing for territory and competing for food. Within and on your body, you are supporting an ecosystem of microbes. These microbes are always there. They're competing with each other. They are limiting each other's growth, which is good for you. They're also limiting the growth of incoming pathogens. Now, another thing they do that I haven't talked about yet is they will help prime your immune system. Your immune system is producing antibodies that recognize these foreign cells and might also recognize more intrusive invaders in the future. Now, you might be wondering, well, isn't there one more category of relationship where neither organism is affected? Well, I'll ask you, is that a relationship? Alrighty then, let's move along and take a look at the effects that pathogens have had on human history, and also the history of how we've responded to them, how we've learned about them, how we've learned to control them and limit their spread. Now, I think I mentioned before, if I'm not wrong on this, that most microbes are not harmful. They're beneficial, in fact. I think I covered that. Anyway, there are some that are problematic, and those, of course, are going to be the focus of this course. We'll take a look at some of the outbreaks that have occurred in the past. I'll touch briefly on what's happening now, and then we'll talk about the scientific method. What you're seeing in this engraving are individuals that are suffering from the bubonic plague. They have been infected with a bacterium known as Yersinia pestis. One of the things that this bacterium does is it elicits a strong response from the immune system that causes lymph nodes to collect fluid and swell. And that creates these swellings all over the body known as buboes. In addition to buboes, people that have the bubonic plague may experience necrosis or death of tissues in their extremities. So for instance, the tissue of the fingers may die and go black, and this has led to the alternate historic term for the bubonic plague, the Black Death. The Black Death was of great historic importance, as we'll talk about in a moment, but it's not something that's gone away. There are still cases of these infections that pop up every year. We have three different forms of infection. We have the bubonic form that we've talked about, which is going to present itself as swellings, so these buboes, and perhaps necrosis of extremities. This is the least harmful of the different forms. We have septicemic plague, and in septicemic plague, the bacterium has found its way into the bloodstream and it's spread throughout the body. It's caused what's known as a systemic infection, a system-wide infection. We can have pneumonic plague, where the bacterium has migrated into the lungs. Systemic, so septicemic infections and pneumonic infections are far more dangerous than bubonic infections, although they're all pretty bad. The fact that we have multiple forms of this infection complicates the life cycle of the bacteria and complicates its pattern of transmission. So in certain areas of the world, the bacterium that causes the plague is always present, and it's found within a reservoir of mice. So mice are maintaining a low background concentration of this bacterium. It's spread from mouse to mouse by fleas. The fleas may on occasion transfer this bacterium to other rodents such as chipmunks and ground squirrels, which might be more likely to come into closer contact with humans. Fleas can transfer the bacterium from chipmunks and ground squirrels to humans. The human becomes a host, and generally they will develop bubonic plague. But the bacterium might migrate to the blood and migrate to the lungs. If it migrates to the lungs, now this individual, this person, can transfer bacteria to other people via a brand new route. Now, if they cough or sneeze, respiratory droplets might contain the bacteria that might be breathed in by other individuals, and they might develop an infection that way. The study of how an infectious agent, such as Yersinia pestis, is spread from one individual to the next or from one population to the next is known as epidemiology. And I'm just giving you a brief little introduction into that field of study, but it's something we'll talk about in more detail. It might surprise you to learn that one of the areas you might come into contact with Yersinia pestis is our neck of the woods, Western North America. However, cases here are pretty isolated and uncommon. When most people talk about the bubonic plague or the Black Death, they're talking about the massive outbreaks that occurred in Europe in the mid-1300s. Now, our history books are kind of slanted towards what happened in Europe. They're rather Eurocentric. We don't have good written records of what was going on in the rest of the world at this time. It would appear that the waves of these infections originated within Asia. And there were probably many, many more deaths in Asia than occurred within Europe. But in any case, the plague was carried along trade routes. So we had infected fleas on infected rats on ships, trade ships, that were moving from Asia to India and then on to Arabia and on to Europe, etc. Now, this is still an important consideration today. It took a long time for infections to spread from one region to the other back in the 1300s, but of course nowadays something can travel on an airliner very, very rapidly. What would happen is the ships that were carrying these infected rats would stop at ports. The rats would leave the ships. They would leave with their infected fleas, and that would transfer the disease onto the inhabitants of that region. What you're seeing here is the spread of bubonic plague through Europe year by year. It took a while. I mean, it took a lot longer than COVID has taken to spread around the world. And of course, that relates to the fact that you couldn't move around all that quickly back in the 1300s. If you wanted to go to Paris, you had to walk there or get on a horse. Nowadays, of course, you can travel around the entire planet in a day. So things spread much more quickly. Now, one thing to note here is that there were some areas that were spared from the infection. They're shown in green. This is a portion of history where we had a lot of ongoing wars and a lot of cities were walled off. And these cities recognized the risk and they quarantined. They stopped outsiders from coming in. And that led to their protection against infection. But it's important to remember that there weren't any people around at this time that really had any sense of what was causing this. Was it evil spirits that were being transferred from person to person? The idea that it would be tiny little microscopic invisible entities would be a rather radical idea. In some towns, there were panics that broke out. If someone got the disease, they would be walled off in their home. People would come and they would put boards across the doors and wall them in, and in some cases, burn down the house as well. So this was an unpleasant time to be alive. And in fact, there's a well-known Italian writer, Giovanni, who talked about plague victims eating lunch with their friends and then having dinner with their ancestors, because this was something that, when it reached a town, spread very, very rapidly. He went on to state that the pestilence was so powerful that it was communicated to the healthy by contact with the sick, the way a fire close to dry or oily things will set them aflame. It's an interesting quote because it suggests that he realized there was some sort of agent that was being passed from person to person, and they had to be in close contact for this transmission to occur. The engraving on the right shows what a typical quote-unquote doctor of the period might be wearing as he went out into the neighborhood to deal with sick people and also to aid in the collection of bodies. So you can see he has a very heavy leather jacket on, and that would protect him from fluids that were being released by the sick or by the bodies of the deceased. He's wearing a mask that has a very pronounced beak on it. The beak would be stuffed with rags that would be soaked in pleasant smelling oils, in part to cover up the smell of those bodies. This was an outfit that you would hope you wouldn't see on the street. And more than a few people have taken to wearing this harbinger of death outfit into places like Walmart. But, well, maybe we can use a bit of dark humor. Over the 5 years that the Black Death did its tour of Europe, 25 million people were killed. That was a lot of people back in the 1300s. I mean, it's still a lot of people, but that was 1/3 of Europe's population. The death toll in other parts of the world was probably considerably higher. So this was a major event in human history. Pathogens have shaped human evolution, human history, and human society. They continue to do so, and they will continue to do so into the future. I have no doubt that you know exactly what you're looking at right now. That is COVID-19. That image has probably been burned into your brain. Is COVID-19 as serious or as dangerous as Black Death? Well, no, of course not. But that's for a multitude of reasons. First of all, COVID-19 only has serious health effects on a small subset of the population. Of course, we're trying to protect that subset of the population. We know a lot more about this pathogen than people did about the Black Death pathogen. People didn't know anything about Yersinia pestis back in the 1350s. They couldn't treat the ill. They couldn't do much in terms of preventative measures either. Even if we look at more recent outbreaks, major outbreaks, if we look at the 1918 outbreak of the Spanish flu, people at that point understood some of the concepts of microbiology, but they didn't know the biology of this virus. They didn't know how the Spanish flu virus spread from person to person, and people were quite lax when it came to taking appropriate precautions. We'll talk quite a bit about the precautions we're taking right now with respect to COVID-19. We'll look at its biology when we talk about virology, and we'll look at how it spreads when we talk about epidemiology. Are we doing too little to thwart its spread, or are we doing too much? Maybe this thing is overblown. We will come back to that. We are currently living through a rather unique period of human history. This is the first time that the entire world has come together and worked together to limit the spread of a pathogen. When I've taught this course in the past, I spent quite a bit of time during my introduction talking about the Spanish flu of 1918 and talking about whether or not something like that could happen again. In 1918, an influenza virus spread around the world. It infected about a third of the world's population, and it killed at least 20 million people. The higher estimates are up around 100 million. People have been concerned for a long time that such a virus might come back, and microbiologists and virologists have been waiting for this virus to make its return. Every 100 years or so, we do have something nasty that circles around the world and causes us lots of issues. And this would appear to be that virus. It has, of course, affected everyone. It's affected everything that we've had to do over the last year. COVID-19 and other coronaviruses are fascinating little buggers, and we will spend quite a bit of time talking about their structure and talking about their genetics later in the course. They contain single-stranded RNA, and that serves as the genetic material for the virus. These are the instructions that your infected cells will use to manufacture the proteins that make up new virus. When the virus leaves an infected cell, it steals part of the plasma membrane, and that forms an envelope around the outside of the virus. It helps protect it from your immune system. It's also absolutely required in order for the virus to enter another cell and infect a new cell. Interestingly, though, if coronavirus is outside of the body, it's actually quite vulnerable, and that's because that lipid envelope can be broken down very readily by something as simple as detergent. At the time of this recording, January 13th, 2021, there have been about 92 million confirmed cases of COVID-19 throughout the world and just under 2 million deaths. This gives us a case mortality rate of just over 2%. And what I mean by that is that on average, on a global basis, if a person becomes infected with COVID-19, they have a 2% chance of dying from that infection. Now, of course, things aren't that simple. If you're young and healthy, your chance of dying is much, much lower. If you're elderly and you have coexisting conditions that exacerbate the problem, so you have heart disease or you have respiratory issues, then your chance of dying can be quite a bit higher. The other thing to note is that in 1 in 5 individuals, there are no symptoms. And also, not everyone gets tested. So our confirmed case number is probably quite a bit lower than it should be. So the case mortality rate is probably a bit lower than what I'm presenting here. Now, what are your chances of getting a COVID-19 infection? Well, they're actually pretty low if we look at it on a global basis. There are approximately 7.6 billion people in the world. We have 92 million confirmed cases. That's a little over 1% of the world's population. So 1, maybe 2 in 100 people have been infected. The US is kind of unusual, though, in that about 15% of their population has been infected. I talked about the Spanish flu before. Spanish flu killed a lot more people, and that's probably because it infected a lot more people. So why hasn't COVID-19 spread more? Is it because it's not as infectious as the flu? Yes, probably. That's probably a big part of it. But we may have played a role in limiting the spread of this virus. We almost definitely have done that. And we probably have done a good job of reducing the number of fatalities by reducing the number of people that became infected. And we will come back to COVID several times. And we'll even talk about whether or not you should inject yourself with bleach. Spoiler, you shouldn't. Let's talk about something a little more fun than COVID. Alcohol. It would be remiss of me to discuss the history of microbiology without talking about fermentation. This is something we've known about as a species for a very long time. And it's the earliest form of biotechnology. We have been using yeast to create ethyl alcohol or ethanol from sugar for thousands and thousands of years. Despite us using microbes for thousands of years to create beer and wine and cheese and all sorts of other yummy things, we weren't able to see microbes until relatively recently. Back in the late 1600s, a wealthy man by the name Antony van Leeuwenhoek went to a county fair and he found a man that was selling weird novelty items or toys that could magnify things using a lens. He was quite excited by these little early microscopes. He took them and he used them to look at the garments that he was manufacturing in his factories and make sure that the weave was just right. He manipulated these microscopes. He built several hundred microscopes over the course of his lifetime, and they were very small and very simple. You can see one of them in the photograph on the right. It had a brass plate. Within that, it had a lens, and then it had a specimen holder or pin that you could attach something to. You would hold this up to the light and look through the lens. And despite these microscopes being so simple, they could magnify things about 300 times. That was enough for him to see bacteria. And he's the first person on record to draw bacteria, draw other microbes, describe them, and describe their motions. In fact, he took scrapings from his teeth. He took the plaque from his teeth and he described the different bacteria that were found within the plaque. One day he was rather sick. He had a rather upset intestinal tract and very loose stool, and he took some of that loose stool, stuck it on that pin, looked through it, and saw this. This is Giardia. This causes diarrhea. And he proposed that maybe this little critter was responsible for his upset tummy. He named these little critters that he was seeing animalcules. Now that idea that something tiny, something invisible to the naked eye, can cause a symptom and cause a disease was rather new. He was way ahead of his time. And in fact, it wouldn't be until much later that people started making these connections again. Early microscopes were advanced further by this fellow. Robert Hooke was a scientist, an English scientist, who did a whole lot of stuff. In fact, if you've done any physics, you might have heard of Hooke's law before. Hooke's law describes how springs work. But he took his microscopes and he used them to look at living things like fleas, as you can see, and he drew some rather detailed drawings. He looked at snowflakes. He looked at all sorts of stuff, including the inner bark of cork trees. So you can take that thick bark and you can cut it into cylinders and use it to stopper up wine bottles. But what he did was he took very thin sections of this material and he saw little compartments, almost like little rooms, kind of like cells in a prison. And he named these little compartments cells. That's where we get that word from. Now note that the picture I'm showing you here, the photograph, probably isn't him. This is a portrait that has appeared in textbooks for decades, but fairly recently it was discovered that that's probably another scientist named Jean-Baptiste von Helmont. Another interesting fellow, he actually discovered that photosynthesis produces oxygen. Anyway, I didn't want to lie to you. We actually don't know what he looked like. We're going to end this topic by talking about science and the scientific method. And the reason this is important is because you're going to be doing some science in the lab, and we're going to be talking about some classic science experiments in the lecture. First of all, science isn't really a thing. It's not a body of facts. It's not an organization of people. It is an activity. It's a methodology. It's a way of asking questions and then logically looking for reasonable answers. It comes from the Latin word to know, and it's a logical and systematic search for information and explanations. And one of the important steps in science is asking a meaningful question, a logical question that you can answer by looking at, hopefully, empirical evidence. Empirical means something you can measure. You can measure the pH of a solution. You can measure the temperature of the air. You can measure the length of an object. Empirical versus theoretical. So theoretical is not something you can measure directly. It's something that you're kind of reasoning out in your mind. Now, sometimes in science, it's very difficult to do empirical observation. If you are a nuclear physicist, a lot of figuring out what's going on in the atom involves theoretical science. However, even a nuclear physicist should occasionally smash some atoms together and look for measurable evidence that supports their theoretical evidence. Any scientific endeavor is going to begin with a lot of observation. Scientists are curious by nature. They get all excited when they come across something that's unknown, something that's new. So let's say we have a scientist that has discovered some sort of new phenomenon that they've never seen before. They're going to spend some time watching it and getting to know it, and then they're going to come up with a reasonable, educated, best guess as to what's going on, what's producing what they're seeing. This is a hypothesis. So it's a tentative answer to the question, what's going on here? This is an explanation that's going to be put on trial. It has to be falsifiable. So the reason that you come up with has to be capable of being disproven, and that's really important. It's very difficult to prove something. It's generally fairly easy to disprove something. So what you're going to do is you're going to take your hypothesis and you're going to do many experiments that are capable of falsifying it. And every time you run your experiment and you fail to falsify it, you add support. But it's kind of tricky to know when you can say, well, we've got proof of this particular hypothesis. In fact, most scientists, good scientists, are very careful with the word proof. They'll say that I've run this experiment 100 times now. I've failed to falsify my hypothesis every single time. This is really good evidence that I'm onto something. Another important thing about a hypothesis is it has to be testable. So for instance, some, but not all scientists, would probably state that religion is outside of the realm of science because you can't design an experiment to test the existence or non-existence of God. It's something that's not really testable, especially if God is going to tinker in the results. Another example, can you test the hypothesis that there is no intelligent life in the universe, except maybe on the Earth? But let's say your hypothesis is that there's no intelligent life out there anywhere. How are you going to test that? You could get on your radio telescope and listen for communications, but just the fact that you don't pick up any communications doesn't mean there's no life out there. If life is on the other side of the universe or the other side of the galaxy, you would have no way of testing that hypothesis. Generally speaking, the scientific process goes something like this. You make observations. You notice something interesting that fascinates you. You come up with questions about what's happening, and you come up with hypotheses as to what you think is happening. Then what you're going to do is you're going to use that hypothesis to come up with predictions. So if I'm right and this is what's going on, then if I do this, this should happen. So that's your prediction. Your prediction is a bit different than your actual hypothesis, and I'll give you some examples in a moment. Next, what you can do is you can design experiments to try to disprove, to try to disqualify your hypothesis. And you do lots of those experiments. The more you do, the better. Also, you should design multiple experiments that come at this problem from a different aspect. You can come up with different predictions based on your hypothesis, and you can test those different predictions with different experiments. Then you make your conclusions. Did your experiment falsify your hypothesis? Do you have to go back and tweak your hypothesis, or maybe take a careful look at your experiments to see if you did something wrong? Or if falsification did not occur, then you've offered support for your hypothesis. The other thing is peer review. Scientists review each other's work, and it's not just one scientist that is testing a hypothesis. It should be several scientists that are testing that hypothesis in different ways. And they should be, and generally are, quite critical of each other. Now I should mention that this process isn't always possible. Sometimes we do science just through observation and sort of rational assessment without an actual experiment. In fact, that's not uncommon at all. Think of paleontology, for instance. If you're studying dead, extinct dinosaurs, you can't really do any experiments on them. However, you can still form hypotheses. So for instance, it's fairly well established that the dinosaurs were wiped out by a meteorite impact. And one of the key bits of evidence for that is that all around the world, just above where we find the dinosaur bones, there's a layer of clay that contains iridium. Iridium is a really rare element on Earth, but it's quite common in meteorites. So it's thought that that layer that's found all around the world is there because of this meteorite impact. Now we could hypothesize that a meteorite killed the dinosaurs, and we could predict from that that we should never find the bones of dinosaurs above that iridium layer. And then we could go out and test that just by going out and collecting samples. If we find dinosaur fossils above that iridium layer, we have falsified our hypothesis and we have to rethink things. Even in a field like microbiology, much of the science that's done in the lab is observational and not experimental. So let's say you discover a new microbe. Well, you're going to maybe look at it under the electron microscope and take a lot of pictures. You're going to describe its structure. You're going to sequence its DNA perhaps. So we're not looking at something functional directly. We're not looking at a process that's occurring. We're collecting empirical data. However, for our purposes right now, we're going to focus on experiment-based science, and you will have a chance to do some experiment-based science in the lab. Okay, so for any experiment, we have a few components that should always be present. We have an experimental variable. This is the factor that you're going to change or manipulate, and it should be the only factor that you change or manipulate. So for instance, if I want to know the effect of temperature on the growth rate of bacteria, I'm going to tweak the temperature, but I'm going to keep everything else the same. I'm going to grow all of my bacteria on the same media, I'm going to expose them to the same amount of light, etc., etc. We have an experimental group, and this is a group of organisms, or it could be a group of test tubes with chemicals in them, whatever it is that you're studying, but this is a group of samples or organisms that are being exposed to this manipulated variable. And then we have our control groups, and the control groups are quite important. We always have to have a negative control. This is a setup where we haven't manipulated that one variable, so we can compare our results from the experimental group to the negative control. We may also have a positive control, and this is a bit different and a bit more complicated, and I'll come back to that in just a bit. Another important concept in science is something known as Occam's razor, and this is named after a monk, William of Ockham, who proposed this back in the 13th century. He stated that when you're testing something, plurality should not be posited without necessity. And that's a rather poetic way of saying, start with the simplest answer. Test that first. So we might have an explanation that is very convoluted. In fact, for anything that you observe, you could come up with an infinite number of possible explanations. Many of those would be kind of silly and overly complicated. So for instance, let's say you walk into your kitchen, you flip the light switch, the light doesn't come on. Well, where are we going to start? What are we going to propose as our first hypothesis? Are there fairies that are grabbing the photons and they're not reaching my eyes? That's a bit silly and also untestable. Did an interdimensional rift open up in my kitchen? Probably not. Is there a short in the wall somewhere? That's more reasonable, but still not the easiest explanation. The easiest explanation is that the light bulb is not working. So you might form the hypothesis that the light bulb is dead. And then you would make a prediction, and the prediction would be, if I put in a brand new light bulb, it should work. And you would test that. Maybe that didn't work. So now you have to come up with a new hypothesis. maybe someone has turned off the main power at the service panel. So my hypothesis would be, that's the issue. The power has been shut down to the house. And my prediction would be that if I go down and switch that switch, I can get the light to light up. So I would try that. Maybe that didn't work. Now I reform my hypothesis, and maybe this time I ask myself, is it possible that I didn't pay my electric bill? I would test that one. And it would be kind of a long way down the list before I would start opening up the walls and looking for a short somewhere. And I would never get to the point where I would consider an interdimensional rift in my kitchen. Let's apply what we've just talked about by looking at a few experiments, and I'll start with some hypothetical ones. Imagine that you are a gardener, and every year you grow sunflowers, and you're growing them for, I don't know, the county fair or something, and you want your sunflowers to be as tall as possible this year. You're going to devise an experiment to test whether or not you can grow higher sunflowers, what you should do to accomplish that. So you want tall sunflowers. This is your problem. What should I plant them in? So you've decided that changing the soil will probably have an impact. That's a good place to start. So you're going to devise an experiment. Your experiment is going to have an independent variable. This is the variable that you're going to manipulate. In fact, it can also be called the manipulated variable. Now we're just interested in changing the soil. That's our manipulated variable, the type of soil that we're using. We're not going to change anything else. All of our plants are going to be exposed to the same temperatures, the same sunlight, they're going to get the same amount of water, etc. We also have a dependent variable. That's what's going to change as a result of our manipulations. So this is something you're going to measure. Ideally, you want something that you can measure empirically. And of course, it would make sense to measure the height of the sunflowers at the end of our experiment. We have our experimental group. That's going to consist of the sunflowers that are receiving this manipulation to that one variable, so the sunflowers that are growing in different types of soil. We have our negative control, and the negative control in this case might be just growing a sunflower in the soil that you normally use. So just regular potting soil, for instance. And we're going to compare the other plants to this negative control group to see if there was any changes over what you usually do. Okay, our hypothesis might be that organic matter enhances growth. So maybe, you've walked around, you've looked at different plants, you've visited different gardens, and you've seen that there appears to be a pattern, and you've decided that you think it's the amount of organic material that's responsible for enhancing the growth of plants. Your prediction, if this hypothesis is correct, that plants will grow taller in soil that contains more organic material. Remember, organic means carbon-based stuff, stuff that came from living organisms. Let's say that we run our experiment for a couple of months, and at the end of that time period, this is what we see. We have our plant that's been growing in potting soil. This is our negative control. We have one that's been growing in a mixture of soil and gravel. We have another that's been grown in soil and sand, another that's been grown in soil and peat, and another one that's been grown in soil and manure. And we see that there's differences in height. Pretend that I didn't actually just stretch them in Photoshop. But with these results, you might look at this and conclude that, okay, manure resulted in the best growth. This supports my hypothesis. It doesn't prove my hypothesis, but it supports my hypothesis that more organics cause greater growth. Now, there could be something else going on, though. Maybe something unusual happened to that plant. Maybe your experiment wasn't as controlled as you thought, so you'd want to do this many, many times. Also, was it the organics? Maybe the manure contained a lot more helpful bacteria. So maybe you might want to repeat this experiment, but tweak it this time and sterilize all the soil and all the components before you grow your plants. So realize, in order to provide sufficient support for your hypothesis, you generally have to do an experiment many times, and you have to do it in different ways and even come up with different predictions that you can test. Now, let's imagine that instead of seeing that obvious trend, you saw this. After 2 months, all your plants were the same height. They were all the same height as the negative control plant. You might conclude that changing the soil has no impact, and that might be a valid conclusion. That does happen sometimes. You manipulate a variable and you find out that that variable has no effect. However, it could also be that something went wrong, or that you got an unusual batch of seeds maybe. So perhaps all of the plants got chilled one night and it stopped them from growing anymore. Or maybe you got a batch of dwarf seeds and all the plants grew to this height and couldn't grow any further. Or maybe the pH of your water was off. It was very acidic and it stunted the growth of all the plants. So if you see results like this, which we call negative results because nothing changed, if you see negative results, you should probably question them. It's very, very difficult to prove a negative result. It's rather easy to provide support for a correlation, a positive correlation. If we see it all the time, we can conclude that, okay, there's something here. But a negative result, it's very difficult to provide proof for a negative result. It's just like my example with the aliens. Just because we've never heard from aliens doesn't mean they don't exist. So what you might do in a case like this is repeat your experiment and include a positive control. A positive control will demonstrate that the system you're studying is capable of generating the change that you're measuring. So we're going to show that these plants are actually capable of growing taller. They're capable of varying in height. So what we might do is add a nitrate fertilizer. Remember I talked about before that plants are limited by nitrogen. If we have a positive control where we include fertilizer and the plant grows taller, it shows us that these plants are indeed capable of growing taller. And that provides support for our negative result. If we ran this experiment, we could more positively say that, yes, it doesn't seem to have an effect. Changing the soil doesn't have an effect on growth. Scientists will always have a negative control in an experiment, but they won't always bother with a positive control. The reason for that is, let's say you do an experiment with just a negative control and you see an obvious trend, you see a correlation of some sort, and you see that over and over and over again. Well, there really isn't a need for a positive control. But if you do an experiment, you get negative results, so nothing changes, then you definitely have to go back and do a positive control. Many scientists will include a positive control in every experiment they do, just in case. It's a bit more convenient. You don't have to go back and repeat that experiment. Let's take a look at another example. What does the pancreas do? Well, back in the 1800s, we really didn't have any idea. And there were a couple of Russian scientists that wanted to answer this question. One of the ways to figure out what something does in the body is to remove it and see what goes wrong. And that's what they did. So they took dogs and they cut out the pancreas. And then they watched those dogs to see what the effect would be. Now this is where observation comes in. They were very observant. Other people might not have noticed what they noticed. They noticed that the kennels that contained the dogs without a pancreas attracted a lot more flies. And they hypothesized that the urine contained more sugar because the pancreas is responsible for regulating sugar levels in the body. So their hypothesis was that somehow the pancreas is removing or bringing about the removal of glucose from the blood. That meant that less glucose got into the urine of the dogs and fewer flies were attracted. So here's what they did to test their hypothesis that the pancreas regulates sugar levels. They had two groups of dogs. One group served as a negative control, the other was the experimental group. The dogs that were in the experimental group had their pancreas removed surgically. So they removed the pancreas from these dogs using scalpels rather than Photoshop, And then they collected urine from these dogs after a set amount of time, and they tested for the presence of glucose in the urine using a chemical analysis. They used a reagent known as Benedict's reagent. Benedict's reagent will react with sugar and it will form a precipitate. But what if they took that urine from the dogs that had their pancreas removed and they added Benedict's reagent and they didn't get a color change and they didn't get any precipitate? It could be that they were wrong and the urine didn't contain any sugar. Or it could be that something was wrong with the Benedict's reagent. Benedict's reagent has a shelf life. After a while, it doesn't work as well. So they included a positive control. What they would do is they would take some urine from a normal dog, so from one of the negative control dogs that still had their pancreas. They would add a little bit of sugar to that test tube, and then they would add the Benedict's reagent at the same time that they were adding it to their negative control and to their experimental group. They should see a color change in that positive control. If they don't, they know that something is wrong with their reagent. And this is one of the most common uses of a positive control. It's used to test a piece of equipment, make sure it's working properly, or to test reagents. So let's review the components of this experiment we just talked about. We had our problem, and our problem was, what does the pancreas do? So specifically, does the pancreas play a role in regulating sugar levels? And you do want your question, your starting question, to be well phrased and specific. The experiment consisted of an independent variable, of course, and the independent variable was the absence or presence of a pancreas. The dependent variable was the concentration of sugar in the urine that was collected from those dogs after a set period of time. Remember, of course, that everything else is going to be kept the same. The dogs are all going to get the same amount of water, they're going to get the same food, they're going to get the same amount of exercise, etc., etc. The experimental group would be the group of dogs that had their pancreas removed. The negative control would, of course, consist of the dogs that didn't have their pancreas removed, but remember, we want to keep everything exactly the same. So those dogs were still sedated, they were cut open, the pancreas was exposed, it wasn't removed, the dogs were stitched back up. So they had the same operation as the experimental group. We have to do that sort of thing because it could be that changes in the sugar level in the urine were due to a stress response, perhaps. So we have to keep everything the same, as close as we can. The positive control was the tube of urine that had sugar added to it. So we use that to test the Benedict's reagent. It also tells us for sure that Benedict's still works if it's in urine. The hypothesis would have been that the pancreas lowers blood glucose levels, thereby lowering glucose levels in the urine. And the prediction based on that hypothesis would be that if we remove the pancreas, we're going to see sugar in the urine. Let's take a look at one more example. This one is actually related to microbiology. So what you're seeing here is the Panama Canal. And the Panama Canal connects the Atlantic Ocean to the Pacific Ocean through Central America. Prior to the completion of the Panama Canal by the US in 1914, if you wanted to travel by ship from the eastern coast of North America to the western coast, or vice versa, you had to travel all the way around the southern tip of South America. A very long trip and a rather dangerous trip. Those southern oceans were rather treacherous to ships at the time. So you can imagine there was great interest in building this canal and reducing that trip distance by over 11,000 kilometers. It was attempted many times. There were several attempts by several different governments to build this Panama Canal, this huge, massive engineering undertaking. The French tried in the 1800s, and one of the problems they ran into, not just the scale of the project, but disease. This was an area of dense jungle. There were many issues with yellow fever and also malaria. They tried and failed, and 25,000 workers died of these diseases. The US followed up on this and tried in 1904, and they were successful in large part because they knew they had to deal with those diseases, and they took precautions to do so, and they put in a lot of effort to understand how those diseases were spread. Yellow fever and malaria are caused by blood pathogens. Yellow fever is caused by a virus, and malaria is caused by a protist, a little single-celled eukaryotic organism. They bring about similar symptoms. One of the things you might notice, very obvious in this fellow, is that the skin and the eyes can turn yellow. This is a condition known as jaundice. What's happening here is red blood cells are being broken down by these pathogens very quickly. Those red blood cells are ruptured, and the hemoglobin that they contain spills out, and it has to be collected by the liver and broken down so that it can be removed from the body. It's not broken down fast enough because the liver is being overwhelmed by all this hemoglobin, and some of the intermediate products of this breakdown are yellow in color, and this material builds up in the skin and the sclera of the eye. Now, at the time, no one knew how these pathogens were spread. There was a Cuban epidemiologist by the name of Juan Finlay that suggested maybe these pathogens were spread by mosquitoes. And that was not a popular idea at the time. That was quite radical. And he was supported by a U.S. Army of Engineers officer, Walter Reed, and the two of them together collaborated in showing that mosquitoes were indeed responsible for the transmission of yellow fever and malaria. What you're seeing in this image here is the yellow fever virus at the top and the species of mosquito that was predicted or hypothesized to carry the virus from one person to another. To do these tests, they used human volunteers, and in fact they also experimented on themselves. Two things that you couldn't get away with today. Again, the idea that a mosquito could transfer a pathogen from one host to another was a rather new and rather radical idea. It was thought at the time by most researchers that yellow fever and malaria were spread either through the air or were spread by fomites. A fomite is an inanimate object that can conduct a pathogen from one person to another. So for example, let's say you have an infection. Let's say you have a bacterial infection and you have this bacteria living in your mouth. Well, what might happen is you might drink from a coffee cup and then share that cup with someone else, and they pick up the pathogen in your saliva, which is attached to the mug. That coffee mug would be a fomite. Or let's say you have a skin infection, a fungal infection, and you share a towel with someone, and they pick up that fungus. The towel would be a fomite. So they set out, Finlay and Reed, to test all three possibilities, to test whether or not yellow fever was spread by the air, whether it was spread by fomites, or of course whether it was spread by mosquitoes. And they used a rather simple little setup. Simple experiments are always the best ones. First of all, they had two bunkhouses. One of the bunkhouses contained people that were exposed to fomites. So what they would do is they would go to a local hospital and they would find people that had recently died of yellow fever or were in the process of dying of yellow fever. And they would take their bedclothes. And yellow fever is a rather nasty disease. It causes internal bleeding, it causes you to vomit all over yourself. It's not pleasant. They would take these pajamas that were covered in vomit and blood and other fluids, and they would also collect the bed sheets of people that had died from yellow fever. They brought these back to their experimental setup, and they made these poor volunteers sleep in those pajamas and sleep in those sheets. They had another building, which is the one you're seeing in the diagram, that they divided into two halves. They did this using a very fine mosquito netting. So that allowed air to move from one half of the building to the other, but it didn't allow mosquitoes to move. On one side, part A, they released mosquitoes. that had drank blood from infected people. So they found people in the hospital again that had yellow fever, and they took mosquitoes in a jar, put that on their skin, made sure that they took a good drink of their blood, and then released these mosquitoes into this bunkhouse. On the other side, part B in this diagram, this was our negative control. These individuals were not exposed to fomites, they were not exposed to mosquitoes, but they were sharing the same air with the people that were exposed to the mosquitoes. What they found was that the only people that got yellow fever were the people that were exposed to mosquitoes. Now you might be wondering, how on earth did they get volunteers for this? Because yellow fever can be deadly. Well, they used people that would otherwise be working out in the jungle to help build the canal. So they gave them a choice. You can make next to nothing to go out and work your butt off building the canal, or we'll pay you slightly more and you can be in our experiment. Either way, whether you're out in the jungle or you're doing this, there's a chance you're going to get yellow fever. So they did manage to get quite a few volunteers. And as I mentioned, they even tested this on themselves. They intentionally exposed themselves to infected mosquitoes to see if they were correct. So let's analyze the parts of this experiment. First of all, we have our problem. Our problem is, how is yellow fever transmitted? And our specific question related to that is, do mosquitoes transmit yellow fever? Our experiment consists, of course, of an independent variable. That's what we're manipulating. What we're manipulating here is the exposure. Our experimental group was exposed to fomites or exposed to mosquitoes. We have our dependent variable. The dependent variable is just whether or not they got yellow fever. Our experimental group, again, that would be the people exposed to the fomites and the people exposed to the mosquitoes. Our negative control would be the group that was not exposed to mosquitoes or fomites. But again, everything else is kept the same. These people ate the same food, they had the same regimen, they slept the same number of hours, they drank the same water, and they were exposed to the same air as everyone else. They did not do a positive control, but we could try to imagine how they might have. Let's imagine that they did this experiment and no one got sick. We got negative results. Now it could be, maybe, that the people they used already had some sort of immunity. Maybe they had had yellow fever in the past, hadn't shown symptoms, and had developed some sort of immunity. Unlikely, but maybe that's the case. What we could do is, after the experiment was complete, we could take the people that we used in the experiment and inject them with yellow fever, inject it directly with a syringe, and see if they got sick. That would tell us whether or not those people that we used in the experiment were actually capable of showing the dependent variable, whether or not they could show symptoms of illness. They didn't do that, as I mentioned, they didn't have to, and that would have made their rather immoral experiment maybe a little more immoral. So the hypothesis, of course, was that mosquitoes transmit yellow fever, and the prediction based on that hypothesis would be that if we expose people to yellow fever in mosquitoes, they will get yellow fever. We won't see yellow fever being transmitted in any other way. The other thing they learned was that the mosquito had to incubate the pathogen for a bit. So they found that if they let the mosquito drink infected blood, put that mosquito directly onto a new host, that person, didn't get yellow fever. The mosquito had to sit for a bit, had to sit for several days or weeks, because the pathogen has to complete part of its life cycle in the mosquito. Before we leave science, I think we should define a few terms that are quite often misused. First of all, what is a fact? Well, it depends who you talk to. You probably don't want to talk to Trump about this, but ideally, you don't get to have your own facts. A fact is something that is inherently true. So for instance, Paris is the capital of France. I think everyone would agree on that. At the very least, a fact is something that's supported by extensive evidence. How much evidence is needed? Well, that's where things get tricky, and that's where different people might disagree. What is a law? A law is a relationship, a very specific relationship or ratio. Quite often it's a relationship that can be defined by an equation. F equals ma, force equals mass times acceleration. Very simple little equation that is applicable in very certain situations. If you're familiar with the gas laws, PV equals nRT describes how gases behave. Theory is a word that causes a lot of trouble because theory means something very different in science than it does in everyday parlance. If you say theory when you're talking to a friend, you probably mean that you have a hunch. I have a theory that so-and-so. A theory in science is an expansive model. It's a model that's supported by lots of evidence. This model contains facts and laws. So for example, there is a theory known as the universal theory of gravitation that explains how gravity works. There is the cell theory that we'll talk about in a moment that states that all living things are made up of cells. Not too many biologists are going to question that. What you're seeing here is a theory or model that describes how blood sugar is regulated. It's regulated by the hormones insulin and glucagon that are secreted by the pancreas. And we understand the details of this quite thoroughly. So remember, we talked about that experiment by the Russian researchers that showed how the pancreas was responsible somehow for regulating blood sugar level. Well, we now know a lot more about this, and as we learn more, that becomes incorporated into this theory, into this model. So theories are big things. They are big, encompassing ideas that are very well supported. Incidentally, in the photograph there, what you're seeing are Banting and Best. They continued to work on the pancreas and isolate insulin eventually. They did that at the University of Toronto with some unfortunate and unwilling volunteers. And when I say unwilling volunteers, I'm referring to the fact that Banting and Best went through quite a few dogs to do their research. Okay, so let's take a look at the history of microbiology. We know today that all living things are made up of cells. We can have single-celled organisms that are living things, and then of course we can have multicellular organisms that are made up of lots of cells. But there was a time when it was thought that life could spontaneously generate. It could appear out of the air and out of inanimate objects. That's an idea known as spontaneous generation or abiogenesis. Bio means life, genesis, the creation of. If you see a at the beginning of a word, it means without. Think of the term atheist, for instance. Theist is someone who believes in God. You put the a in the front, it means they don't. Anyway, according to spontaneous generation, there's this vital force in the air, and it can breathe life into inanimate objects. The alternate hypothesis, which is the one that we follow today, is known as biogenesis. Living things come from pre-existing living things. You came from your parents. You didn't simply poof into existence by the air breathing on, I don't know, piece of cake or something. Who knows? We have what we call now the cell theory, and this has been around for a long time, since 1839, and it was developed by Schleiden and Schwann. So this is an example of a theory, a model, that is very, very well supported. Up until Schleiden and Schwann, that's not right, Schwann came along, the idea of spontaneous generation was widespread, not just among the general public, but among scientists, among biologists as well. And in fact, if you go back and look at papers in the 1800s and 1700s and further back, you can find all sorts of recipes for making different life forms. Let's say you needed mice for some reason. There was a recipe for that. What you would do is you would get yourself a large open-mouthed jar. It had to be open, that's pretty important. You put that in a barn. You also throw in some wheat and some underwear. Some recipes suggested socks instead, but it seems to be rather important that the underwear or socks be dirty. Now, the reason the jar had to be open, of course, is so that air could get in and breathe that vital force into the underwear and the wheat. And if you left this in the barn overnight, the next morning you would come in and sure enough, you would have mice. This sort of thinking seems rather bizarre and rather silly to us today. And I'm sure if you invested a second or two, you could figure out a more rational, simpler explanation as to how those mice ended up in that jar. But remember, this was a widely held belief. And I should also point out that there was at least one moment in history where life did appear, although it appeared over hundreds of millions of years. Anyway, there were some early experiments that attempted to disprove spontaneous generation. And the earliest of these, at least the earliest that we know of, was performed by Francesco Redi. And Redi did the very, very first proper controlled experiment that we have records of. So that's a pretty important event. He also had some pretty cool hair. Okay, what he did was he demonstrated that maggots do not spontaneously appear from meat. So a maggot is the larval form of a fly, just like a caterpillar is the larval form of a butterfly or moth. Here's the life cycle of the fly. We have a maggot up the top, which is a little worm-like organism that burrows through decaying meat and other things, and then it pupates, which you can see on the right. It hatches out as a fly, and then the fly lays eggs, and then the eggs hatch out into maggots. He did a very simple little experiment. So people at the time thought that if you took meat and let's say you left it out on the counter in your kitchen, you would eventually end up with maggots. And the reason they thought that was occurring is because vital force in the air was breathing new life into the meat, and these maggots were spontaneously generating. So he did a few simple tests. He took some flasks and he put meat into these flasks, and he left them out on a countertop. He had one flask that was unsealed. So the meat had exposure to the air, and he did indeed observe that maggots developed in that meat. He had another flask that he sealed tightly with a cork. No air could get in, no maggots appeared. Now, if you were a believer in spontaneous generation, again, as most people were at the time, you would say, well, of course not. The air can't get in, the vital force can't get in. But then he did a third experimental setup where he put cheesecloth over the top of this jar. No maggots appeared in the meat. And in fact, he was an observant fellow, and he noticed that flies were trying to get to that meat, and in fact they were laying eggs on top of that gauze, but the eggs couldn't get to the meat. And this is something really important. I've stressed this a few times, but if you're a good scientist, you are making observations all the time. He had done something and recorded something that you would have thought would have been done a long time ago. He actually just watched the flies, and he saw the flies landing on the meat and laying eggs, giving rise to maggots. These sorts of experiments continued. One by one, people showed that spontaneous generation was not occurring. But there were a lot of holdouts. So sure, you've showed that maggots can't spontaneously generate, but what about hornets and wasps? There was a recipe where you took a dead ox, buried it in the soil up to its neck, came back a few weeks later, and you'd find hornets feeding on it. Well, of course, they didn't think they were feeding on them. They thought that they had spontaneously generated. So one by one, though, tests were done that disproved spontaneous generation. The last holdout was microbes. So once we started studying microbes and looking at them under the microscope, these were tiny little simple things, and it was thought well into the 1800s that microbes could spontaneously generate. And it was Louis Pasteur that put the final nail in the coffin of spontaneous generation by showing that that was not the case, even for tiny things like bacteria. Louis Pasteur was an excellent scientist. This is one of his quotes here: "Do not put forward anything that you cannot prove by experimentation." So everything for him had to be demonstrated by empirical experimentation. This is Louis Pasteur's classic experiment where he finally put an end to thoughts of spontaneous generation. This is an important experiment, as was Reddy's experiments that you might want to know about for, a test or something. Hint. Anyway, what we've got on the left is a long-necked flask. And what Louis Pasteur did is he put beef broth, soup basically, into this flask, and then he would take that, put it over a Bunsen burner, boil it, sterilize it, and if he took that flask and he let it sit, eventually it would go bad. And that's because bacteria would fall into the flask, and the spores of bacteria would fall into the flask and give rise to a population of bacteria that would spoil that beef broth. And if he took some of that and put it under the microscope, he would see that there were bacteria there. But he did another little test. So doing something like that would be his negative control. He hasn't done anything. He hasn't changed anything. But some of the flasks, he took them and he took that long neck, heated it up so that he could bend it into a swan neck. We call that a swan neck flask. He would boil that solution and let it sit, and he could let that sit for weeks and even months. The solution never went bad. It never became contaminated with bacteria. And the reason for that is, imagine that we have bacteria in the air and we have spores of bacteria in the air, and they fall down onto that spout, and some of them might make their way down to the bottom of that spout, but it's a long way to go to get to the solution. None of those bacteria, as tiny as they are going to make their way to the solution and cause it to spoil. So this satisfied all the concerns of people that believed in spontaneous generation, because air, of course, could get to the solution, the vital force could get to the solution, but the bacteria physically settled out in that swan neck. So once again, he was able to show through this that spontaneous generation was not a thing. So this simple experiment by Pasteur cast extreme doubt on spontaneous generation even within invisible microbes. He had his experimental setup, which consisted of broth that had been boiled within a swan-necked flask. His independent variable was whether or not bacteria could reach the broth. So that swan neck prevented bacteria from reaching the broth, but allowed air to reach the broth. He had a negative control, which I haven't discussed, but basically he took a flask and boiled the broth to kill any bacteria that might be there, and then he sealed the flask, so neither air nor bacteria could enter the flask. And then he had his positive control, where he had a flask that was wide open, and of course, whether you were a spontaneous generationist or not, you would agree that bacteria could get in, and also air, the vital force, could get in. Now, Pasteur is known for a number of other things, and we'll come back to Pasteur later. He worked for a number of wineries, and that's how he made quite a bit of his money, trying to improve the process of fermentation. He showed that fermentation was caused by microbes. It was caused by yeast. He also came up with a process that you're probably familiar with, pasteurization. It's named after him. And this is where we can kill spoilage bacteria by heating a solution for a very short period of time. So we're using high heat for a short period of time. The period of time is short enough that we're not changing the properties of the liquid or the food that we're pasteurizing. he also showed that rabies was not caused by a bacterium. And this is rather interesting. So up to this point, we had identified single-celled eukaryotes. We knew of their existence in any case. We knew that bacteria were a thing, and Louis Pasteur was one of the people that showed that bacteria and other microbes were responsible for disease. But he noticed that rabies was caused by something smaller. Basically, he found that he could take fluid, bodily fluids, from an animal with rabies, inject them into another animal, and give that animal rabies. The interesting thing was, if he took that fluid and passed it through a very, very, very fine filter, a filter that was fine enough to filter out bacteria, that filtered fluid would still cause rabies in the other animal. So he realized that something smaller than bacteria must exist that is capable of causing disease. We know now that that's a virus, but of course this is not something he was able to see with the equipment that he had available at the time. Observations and experiments performed by the French biologist Louis Pasteur and the German physician Robert Koch led to the development of what we now know as the germ theory of disease. This is the idea that microbes cause infectious diseases. Again, theory, not a hunch. This is a well-supported model. Infectious diseases are not caused by witchcraft or evil spirits. They're caused by little tiny microscopic infectious agents. Robert Koch was one of the first people to show definitively that a specific infectious agent caused a specific set of symptoms. He showed that a very specific bacterium caused anthrax. Koch realized that if we're talking about a disease, that disease has a certain set of symptoms that define it. There was a certain set of symptoms that defined anthrax in cattle or another ungulate. And if we were looking at an animal with anthrax, we would always find the same specific bacterium. This is a bacterium that he identified and later became known as Bacillus anthracus. He gave us a protocol that we could use to link diseases to certain organisms. And that's a set of protocols that's now referred to as Koch's postulates. Koch's postulates are still used today. In fact, they still form the core of investigative pathology. Imagine a situation where we have a brand new infectious disease popping up. Hard to imagine, but let's say that that is what happens, and we want to figure out what particular microbe is responsible for this set of symptoms. In this example here, we have some lab mice. They're showing the same symptoms. We want to figure out what's responsible for that disease. So what we're going to do is extract a sample, maybe some blood, maybe other fluids or tissues. We're going to extract this sample, and it's going to contain probably lots of different microbes of different species. Only one of those is going to be responsible for the disease we're interested in. The other ones might not be doing anything at all, or they might be causing other problems. But we want to isolate out the microbes and grow pure cultures. So that mixture that we extract from the animal, it's a mixed culture. It contains more than one species, most likely. What we can do is we can take that mixture and we can take it and spread it out on a petri plate. And let's say we're dealing with bacteria. Well, individual bacteria cells are going to separate out, settle out on top of that petri plate, and they're going to give rise to colonies. And we might see several very different looking colonies. Some might be yellow, some might be white. They're going to have different textures. They're going to look different because those different colonies are made up of different bacteria. Where one bacterium settled out, it will grow over and over and over again until we have a mass of billions of bacteria that were all derived from the original bacterium that settled out there. Now we can pick up one of those colonies, transfer it to a new tube, and grow a pure culture that only contains one species of bacterium. And it's really important that we get a pure culture. Now we're going to take those pure cultures, we're going to make slides, we're going to look at them under the microscope, we're going to make note of the biochemical characteristics of the bacterium, we're going to make note of the shape of the bacterium, we might even isolate DNA. We're going to characterize each of those species that we've isolated. Now what we're going to do is we're going to take a pure culture and we're going to inject it back into a healthy mouse. And we're going to see if we can replicate the set of symptoms that we started with. If we have several different pure cultures of different bacterial species, we'll have to do this several times. But we're going to find the microbe that causes the original set of symptoms. So we're going to infect our new host. We'll look to see that we get the same symptoms. That's important. And if we do, we will once again extract some fluid, look at it under the microscope, perhaps grow up the bacterium first so we have more to work with, but take a look at that bacterium under the microscope again, run the same test that we did before, and see if we have the same species. So to recap, we have a set of symptoms in a particular organism. We're going to isolate microbes. We're going to grow up pure cultures. We're going to take those pure cultures, re-inject them into a new healthy host. We're going to find out if that host gets the same symptoms as the mouse that we started with. And then we're going to look, and this is very important, this last step, we're going to look to make sure that the second time around, we have that same microbe again. If all of that works out, then we have really good evidence that we have associated a particular pathogen with a particular disease, i.e. a particular set of symptoms. A little while before the development of the germ theory and Koch's postulates, we had other people that were realizing that there's something that can be transferred from one person to the other, even if they couldn't characterize it. So in 1847, for instance, a man by the name of Ignaz Semmelweis realized that if you washed your hands, it made a big difference in terms of people getting sick. He worked in a hospital and he realized that if people washed their hands as they went from one patient to another, they were less likely to affect the spread of a disease. Now, once again, he did not know what was going on. He didn't have the information that was available to Koch later, and it was quite a while before people realized the significance of these sorts of observations. But eventually the germ theory did become incorporated into how physicians operated. Pardon the pun. So for instance, Joseph Lister, who you might recognize, he's the inventor of Listerine, he realized that if we were to limit the growth of microbes, following the proponents of the germ theory, then we could limit infection. So back in Joseph Lister's day, if someone had to have a surgery, if they had to be cut open, there was a good chance that the surgery was going to be worse for them than the actual ailment they were trying to fix, because there was a very good chance of serious infection. What he would do is, as you can see, he would have these little stools set up and he would have these little atomizers on top of them, these sprayers that would spray out a rather harsh solution that contained phenol and some other nasty things. And these aerosols, these tiny little droplets, would fill the room and destroy, hopefully, any bacteria or spores that might be drifting around in the air. It probably made it rather difficult to breathe for the attending physicians as well, but it proved to be very, very helpful. It proved to cut way back on infections on the operating table. Listerine, the original formulation, contained phenols and some rather nasty compounds. It has since been tamed a bit, but it does trace its roots back to this man. Another rather important development in the history of microbiology was the concept of vaccination and the understanding of immunity. Immunity refers to the fact that if you're exposed to a pathogen and then exposed to it again a second time later in life, the second time around you are at a distinct advantage. you can launch an immune response more quickly because your immune system has a memory of sorts. And this is something we'll talk about in much more detail later in the course. Of course, vaccination is something that's in the news right now for obvious reasons. Edward Jenner realized that if someone was exposed to the cowpox virus, it granted them some immunity to the much more serious smallpox virus. He was a wealthy individual, as were most scientists and researchers of the day. He had a lot of spare time on his hands, and he had a lot of servants that he could use as volunteers. He famously took the cowpox virus and injected it into the son of one of his servants, and then exposed this boy to smallpox. And the boy did not get smallpox, which is good because smallpox can be quite deadly. Cowpox, which is something that generally just results in a mild rash and maybe a little bit of nausea, is quite common in that day in people that were working around cows, and he saw it in his maids that were milking cows. The name vaccination comes from the word vacca, which is Latin for cow. From 1857 to 1911 or so, we have a period of time that's often referred to as the golden age of microbiology. Now, this is not a history course, and I will never ask you for dates, and I'm not going to ask you to give me tons of information about the personal lives of these people, but there are a few people that I do want you to know about. I want you to be able to tell me in just a couple sentences what their contribution was. Of course, you should know more about Koch with respect to his postulates. You should be able to describe those, and you should know a little bit more about Pasteur and his experiments that disproved spontaneous generation. But you can see here, Pasteur pops up quite a few times in this timeline. Lister, we've talked about him. Koch, what other names should we look at? Gram, we'll talk about the Gram staining procedure quite a bit. That's someone we'll get to later. Escherichia, who gave us the name Escherichia coli, which is kind of fun to say. Petri, which came up with the Petri dish. Actually, his wife invented that. We'll talk about that later. Ehrlich, who invented a number of stains and things that we'll use in the lab, and also did a lot of work on immunity. We'll come back to some of these people a bit later in the course. Microbiology has had a huge impact on human history. In fact, I suspect most microbiologists would tell you that it's the most important science out there when it comes to its influences. Just looking at this graph here, you can see that we've had an overall drop in the rates of infection. So you're seeing death rates per 100,000 people on the y-axis, and you're seeing the dates from 1900 to 2000 along the x-axis. Things like the establishment of public health departments, which told people, maybe you shouldn't be dumping your garbage and your sewage on the street, had a big impact. The chlorination of water had a big impact. The development of vaccines, big impact. And these are things that continue to be important. There's an interesting spike there, which is the Spanish flu pandemic. And we'll come back to that. Having said that, we have seen the resurgence of some diseases, and in some cases they're coming back a bit more virulent than they were in the past. And we're also seeing the evolution of brand new forms of pathogen. One of the things that is responsible for the faster outbreaks of disease that we see today is the fact that there are a lot more of us. I mean, for a very long period of time, for most of human history, there were only a few million people on the planet. And of course, now we have close to 8 billion people. We can also travel around the world very quickly, which means things can spread more quickly. So despite our huge advances in medical science, we have to really carefully analyze the epidemiology of these new diseases. So here you're seeing the growth of human population. It's thought that it will level out perhaps A lot of estimates say around 2050, which is a good thing because we've been having some extreme exponential growth for some time. It's interesting in this graph, you can even see the effect of the Black Death. Yowzers, that was a long topic. I have a few concepts here which I'll run through very, very quickly. Microbes are everywhere, they're amazingly diverse, and they're amazing in general. Microbes are hugely important to us. They do all sorts of important things, and we wouldn't be here without them. So again, don't get overly scared of microbes. Most of them are not going to kill you. Microbes have repeatedly shaped the course of human history, and they will continue to do so, and of course they are doing so right now. Science is the process of logically and systematically testing a hypothesis, and you should understand the scientific method and how you apply it. On a test, for instance, I might give you a hypothetical experiment and ask you to identify the different components, or I might ask you to design a hypothetical experiment. A hypothesis must be testable and falsifiable. That is important, and we always start with the simplest hypothesis. If we need to create something more convoluted and complex, we can do that, but only if we have to. Scientific theories are comprehensive. They are models. They are well supported. They are not hunches. Louis Pasteur, Robert Koch, and numerous other people made significant contributions to our understanding of microbes, and we will come back to some of them later in the course. Again, though, this is not a history course. And here's our terminology list. Considering the length of this topic, it's fairly short, and that's because we will come back to some of these concepts again, and we'll definitely come back to a lot of the microbes and pathogens that I've talked about, and we'll revisit them. Now, as always with my courses, when I'm writing my tests, I have the terminology list in front of me, and that's what I draw from. So you should be able to define these terms and use them and be able to explain them to someone else. Explain them to your parents. If they understand what you're talking about, then maybe you understand the terms too. But you can expect to see these terms referenced in matching questions, true, false, multiple choice, short answer, etc. There are a few people I would like you to know about. This isn't a history course again. I don't expect you to know their first name even. If you just know their last name, that's fine. I don't expect you to know when they died, when they were born, when they made their contributions. Don't worry about dates, but be able to describe their contribution in 1 or 2 sentences. There's a few of them that you should know about in a bit more detail. For instance, Robert Koch, you should be able to describe and apply Koch's postulates. For Louis Pasteur, you should be able to tell me about his experiment that disproved spontaneous generation. For Francesco Redi, you should be able to tell me about his experiment that also disproved spontaneous generation, at least in flies. Finally, here's a few broad study questions, a few things for you to consider. Now, I have supplied you with a study guide as well. Remember that your study guides are not comprehensive. The study guides just contain kind of the big concept stuff, but I give them to you as Word files because I expect you to take notes and I expect you to flesh out those study guides a bit more. Likewise, you have the raw PowerPoint of this. I encourage you to add your own annotations, make your own study guides, make your own flashcards, whatever is helpful for you. And that's it for this week. I will talk to you again next week.